Hi everyone! Today's video is the first of a series about human embryology, or how a single cell gives rise to a human being. This video's topic is the first week, which goes from fertilization to right before implantation into the uterine walls. Enjoy! The start of the first week of human development is marked by fertilization, for which a single sperm comes in contact with the oocyte. Fertilization takes place in the ampulla of the uterine tube. On their way to this location, most sperms die or get lost. In fact, it is estimated that a little more of 1 out of 1,000,000 survives the journey. Fertilization takes place in 3 major steps. Penetration of the corona radiata, a layer of granulosa cells nourishing the oocyte. Penetration of the zona pellucida, a thick glycoprotein coat covering and protecting the oocyte. These glycoproteins allow the sperm to recognize the oocyte and, being highly specific, to prevent interspecies fertilization. Both penetration of the corona radiata and the zona pellucida are made possible by lytic enzymes contained in the acrosome, a cap-like vesicle on the sperm's head. Contents of the acrosome are released by exocytosis when approaching the oocyte, a process called acrosomal reaction. Only capacitated sperm can undergo acrosomal reaction. The last step is the fusion of the sperm membrane with the oocyte membrane and the entrance of the sperm's organelles and the nucleus inside the oocyte. As soon as the sperm and oocyte fuse, 3 more events occur. The oocyte releases cortical granules, which induce a hardening of the zona pellucida, acting as a mechanical barrier against other sperms trying to fertilize the egg. The oocyte, which was stuck in metaphase 2 until now, resumes and completes meiosis 2. This division yields a small haploid cell, called second polar body, as a byproduct. All organelles of the sperm, except for the nucleus, are degraded. To recap, the sperm and the oocyte are fused together into a single, diploid and totipotent cell. This cell is called the zygote. If a second or possibly more sperm manages to fertilize the same egg, polyspermia occurs. The extra DNA provided by this additional sperm causes triploidy of the zygote, that is, the zygote has 3 complete set of chromosomes. Triploidy is not fit for survival at all and results in spontaneous abortion early in the first trimester. Right now, the zygote is a cell with 2 pronuclei, which are 2 nuclei with each a haploid set of genetic material that flows freely in the uterine tube. The male pronucleus contains the DNA of the sperm, while the female pronucleus contains the DNA of the oocyte. Soon, both pronuclei duplicate their DNA and merge together. Then, the zygote undergoes its first mitosis. After that, this 2-cell zygote undergoes many more mitotic divisions in a process called segmentation. At each cell division, the number of these cells, called blastomeres, doubles, but the size of each single cell halves. This is because the zygote is still limited by the tough zona pellucida and it cannot grow any larger. Initially, blastomeres are loosely arranged beneath the zona pellucida, approximately at the 16-cell stage. Though, blastomeres undergo a process called compaction. They come closer to each other and maximize their surface contact. Then, cleavage resumes and the blastomeres go on dividing. Following compaction, we end up with some cells on the surface of the zygote and some others segregated deep inside. The former are called the outer cell mass, while the latter are the inner cell mass. This step is really important, but we'll come back to this in a few minutes. Now, the zygote is called a morula because it vaguely resembles a mulberry. By the time the morula reaches the uterus, a fluid-filled cavity called the blastocele starts forming inside it. This cavity is formed as the morula actively pumps sodium ions inside, and water follows due to osmotic pressure. As the blastocele grows larger and larger, the morula takes the name of blastocyst. No water, though, is pumped inside the inner cell mass, which remains segregated at one pole of the blastocyst. The inner cell mass, now called the embryoblast, will give rise to all the tissue of the embryo proper. The outer cell mass, now called the trophoblast, plays a key role in interacting with the environment and will give rise to the fetal placenta. Segregation of the embryoblast and trophoblast is a very important step because While before all cells of the zygote could give rise to any tissue they wanted, now cells of the embryoblast are tied to give rise to embryonic tissue, and cells of the trophoblast to extraembryonic ones. So, this step marks the loss of totipotency. Roughly at the same time, the blastocyst gets rid of the zona pellucida. From now on, functions of the zona pellucida, such as protection, will be carried out by the trophoblast. Without this process called hatching, the blastocyst couldn't get any bigger than this, and even more importantly, implantation into the uterine walls wouldn't take place.